We're back again with the culmination of our conversation around central dogma and to talk about the genetic code. And we'll talk a little bit more about translation here. So let's take a peek at some of the properties of the genetic code. The genetic code is composed of triplets, that is, they are triplet codons. It is non-overlapping, that is, each of the triplets is read in order by the ribosome. The genetic code is comma-free. It is degenerate, ordered, obviously, but it does contain start and stop codons, so there is some punctuation. The genetic code is nearly universal, that is, all living things have basically the same codon table, that is, the same codons code for the same amino acid. So we can see that there may be some differences between some prokaryotes or even archaebacteria and eukaryotes, but certainly if we're just talking about eukaryotes or prokaryotes in general, for example, E. coli, the majority of the coding is identical. And this is really handy when you're doing things, for example, our transgenic laboratory where we're inserting the code for green fluorescent protein from jellyfish into E. coli. And of course, E. coli synthesizes the fluorescent protein perfectly well. So it can, the bulk of the code rather, actually does in fact translate very well across all living things. One thing to note is that you can see here's another look at our at central dogma. We have DNA transcribed to messenger RNA translated to a polypeptide chain. Ultimately, if we take a look at the DNA, notice a couple things about it. Notice that we always, and in fact you must, when answering questions on any of the exams, you must label the 5' and 3' ends of DNA. Notice in this case, of course, DNA has two anti-parallel strains. That is, the upper strand goes in the 5' to 3' direction, meaning the lower strand is 3' to 5'. Notice that the messenger RNA is transcribed in the 5' to 3' direction. Why? Because RNA polymerase rides along the template strand in the 3' to 5' direction. So in this case, the lower DNA strand, 3' to 5', is indeed the template. And you can tell because the messenger RNA sequence is basically the same as the upper sequence of DNA, the coding strand, right? It's the coding strand, and you can tell that because the nucleotide sequence is identical with the exception of uracil replacing thymidine. So very easy to see. Notice that the triplets stay the same between the DNA and the messenger RNA, and each triplet is read in order, in sequence, to be translated by the protein synthesis apparatus, the ribosomes, into a peptide. Notice the peptide goes arginine, glycine, tyrosine, threonine, phenylalanine, alanine, valine, and serine. Again, get used to seeing the three-letter codes for the full name of the amino acids. I should point out that just as when you are writing DNA or RNA, you should always indicate the 5' and 3' ends. When you are writing a peptide chain or a protein chain, you should always indicate which end is which. That is, one end will have the amino or NH2 terminus, and the other will have the carboxy terminus or COOH. In this case, it's good to remember that the 5' to 3' messenger RNA gets translated into the amino terminus to the carboxy terminus. So in this case, the arginine is the amino terminus, so you would write this NH2-ARG. You'd fill in all the other amino acids. And of course, the final amino acid is serine, and you would write it SER-COOH. And that way you can never get confused about the orientation of the gene, the messenger RNA, and the peptide chain. So it's really a standard way to annotate sequences. So especially as a beginner, you always have your orientation amongst the molecules correct. There's always a trick question on exams where someone will write, instead of writing 5' to 3', they will write 3' to 5', which means you have to create the complementary 5' to 3' strand to create the next messenger RNA and, of course, the protein. So don't forget, you have to pay attention to the orientation in the molecule. Of course, some interesting things happen with DNA, and we'll talk a little bit more about mutation as we go along. Here's a simple DNA sequence. Hold on one sec. Okay, sorry about that. I forgot to silence the fax machine. Now notice in the original DNA sequence, we've already got it separated into triplet codons for you. Of course, there are no spaces in the actual DNA molecule. And notice what happens if we have a mutation where there is a single base substitution. So in this case, we go from that center triplet, GTA, to GCA. Importantly, while GTA and GCA may code for different amino acids, notice that the subsequent codons are unchanged. That is, we still have the final two codons being GAT and CGT. On the other hand, if we have a base insertion, meaning here a thymidine residue is placed where the A used to be, now we have GTT and then AGA-TCG. And in this case, the subsequent triplet codons themselves change because, since ribosomes interpret the message in triplets, it doesn't know where the punctuation should or should not have been in the messenger RNA. By the same token, you can see if we have an individual deletion that occurs, our original GTA, removing the T, becomes GA, and it reads the next G from what used to be another triplet. And you can see that all the subsequent codons are also changed. And we call this a change in reading frame. And that is the manner in which DNA is read. So if you take a peek, think for a moment. Here's some thinking questions. In a given nucleotide sequence, how many reading frames are there? So you have to think to yourself, how does RNA polymerase create a message? It moves in a 3' to 5' direction. So along one strand of DNA, it can move from the 3' to 5' direction, but it can also be on the other strand moving in the opposite direction, still in the 3' to 5' direction. So both strands of DNA can be used as a template by RNA polymerase. So let's take a moment and take this DNA sequence. Here we've got 5' ATG, ACC, GTG, GGC, TCT, TAA. And if we were to convert these into the individual amino acid components for each triplet, you would see we get methionine, threonine, valine, glycine, serine. They're those single letter codes. Of course, we could just as well begun the triplets at the letter T, so at the second nucleotide. And if we were to do that, notice we get a TGA, CCG, so completely different set of triplets, resulting in a completely different amino acid sequence. A completely different amino acid sequence. And what if we went to the G, or the third nucleotide in the given sequence? Well, then GAC, CGC, T, GGG, etc. And notice a third completely different sequence. Of course, if we went to the fourth nucleotide in that sequence, well, we would just start with A, right? Because if we count A, T, G, 1, 2, 3, A is fourth, well, then it would be ACC, GTG, GGC, and it would basically be the same as the first sequence, except minus that pesky little methionine residue. So there are 3 forward reading frames. How many are backwards, meaning in the alternative strand? Right, there are 3 in the other direction. And of course, you should be able to figure out what the 3 backward reading frames are. Of course, that means you have to make the 5' to 3' DNA sequence from the complement of the single strand given in our example. And I'll leave that to you to write out at a later date. In general, if we're talking about a random sequence or an open reading frame of DNA, a stop codon generally follows methionine, which of course is our start codon, AUG, in about 20 amino acids or so. Generally, much longer open reading frames are within genes or in terms of even parts of genes. So we see some really interesting differences in the eukaryotic genome with regard to how much DNA is actually converted into protein. So just to put some names on this idea, if we have a sequence of DNA, notice we've got the sequence of DNA being the bottom, in this case, even though it's not labeled by the textbook, the upper strand of DNA under the no mutation box would be our 5' to 3' coding strand. The 3' to 5' template strand is below, which is AGT. The messenger RNA matches the coding strand, even though it's created from the template strand, and codes for serine. Now, if we have a mutation that results in a different amino acid, so in this case, the mutation creates a messenger RNA that's UUA, which codes for leucine, we have what's called a missense mutation. That is, that there's still an amino acid there, but it's not the same amino acid that was in the wild type or non-mutated DNA. We could also have a nonsense mutation, and in this case, notice we went from TCA to TAA, which created a UAA messenger RNA. This happens to be a stop codon, so protein synthesis is terminated, and notice that we have a much, much shorter protein or peptide because protein synthesis stopped prematurely because of the mutation. This is called a nonsense mutation. Nonsense mutation. Finally, there is also the possibility, if there is a mutation, and these are very commonly occurring in the third nucleotide of a triplet codon. So instead of TCA, it becomes TCG. It turns out that the codon UCG also codes for serine. So we have the exact same amino acid sequence, but obviously a different genetic sequence. So this is called a silent mutation. That is, there is no obvious phenotypic difference because the protein sequence is the same. So this is really, and this has important evolutionary implications, which we'll talk about a little bit later. Here, of course, is your entire codon usage table. This is the classic way. I happen to like this one because it's color-coded. Notice that the first letter of the triplet codon is along the leftmost column. Then the second letter is across the topmost column. And then the third letter is sequentially placed within the box. And so you can kind of see each of these codes, for example, UUU codes for phenylalanine, which is indicated by the single letter code F, okay? Notice that if you do your math very quickly, how many total possible triplet codes are there? Well, there are 4 nucleotides, 3 positions, right, in the triplet, so it becomes 4 times 4 times 4, or 64 total possible combinations. How many amino acids are there? Think for a minute. 20. So it means that more than one triplet codon codes for the same amino acid. And you can see, for example, with leucine, in the CU codon, all 4 possible triplet codons, CUU, CUC, CUA, and CUG, all code for the same amino acid leucine. There are a couple that you should pay close attention to, and that is these special, what I'll call punctuation marks, and that is AUG. It is the only triplet codon that codes for methionine, and that is the start codon, the start codon. So all messenger RNA is translated at the beginning of an AUG. There are, however, 3 different terminators or stop codons, and they are indicated by UAA, UAG, and UGA. These are the 3 most common of the terminators. All right. Now, all of these were figured out and given by Khorana, Nirenberg, and Holley, and actually they were awarded the 1968 Nobel Prize. Remember, Watson, Crick, and Wilkins got their Nobel in '67, and ultimately Khorana, Nirenberg, and Holley were '68. Nirenberg actually figured out the first of the triplet codes that coded for proline, and then ultimately it was Khorana who filled in the rest of the table. So really a tremendous advantage for us. To me, the absolute most fun that one can have is taking DNA sequences, probing the DNA sequences for genes, and trying to identify what the protein product is of those genes. It's really quite a bit of fun, and obviously you can use computers for this. You don't want to do, for example, a 300 or 400 amino acid protein by hand. So we use computers to do this, and of course that adds an element of fun as well. One final concept about the triplet code, and this is just another copy of the same codon usage table that we saw in the previous slide, is this concept of wobble. And wobble shows that really changes in the 3rd position are the changes that are most likely to result in the same amino acid being coded for by that piece of DNA. And if you look through the table, you could see, for example, UCU, UCC, UCA, UCG all code for serine. It is the 2nd column to the right at the top row of the table, and it's called wobble. There is a great video in the next one in this Piazza post which will show you a graphic demonstration of how wobble might work. And it has to do with exactly how the transfer RNA binds to the ribosome to bring in that amino acid. And so you'll get a chance to see that. And of course, here we see the difference among and between all the different nucleotides and their position and the anticodon, and of course what the third position of the codon is. So really nice example, great table by Pierce in order to show you that. Ultimately, this is one of my favorite codon usage tables, and it is actually in a circle. So you can see in the middle, for example, here's U, U, and U. It codes for phenylalanine. There's the F. It gives you the molecular weight of phenylalanine and ultimately even shows you the structure. And it is color-coded to show you that phenylalanine is actually nonpolar, so all the yellows are nonpolar. And of course, each color has its own. I just happen to like this. In my old office back at Penn State, I used to have a very large poster of this in my office, and it was one of those things you could really ponder over a little. Very handy to have, to be able to look at and understand some of the coding factors, and of course try to identify some of the properties of peptides that are coded for by a particular gene sequence. Here, of course, is an example, and we will talk much more about this in the future. Here is a codon usage table, and in particular annotating individual diseases as a result of a change in the particular mutation. So if we were to look, we started out looking at sickle cell disease, and in fact, if you look down here at the bottom, there's a green arrow that moves from right to left in the bottom row. And what does happen in sickle cell disease is a GAG that codes for glutamate or glutamic acid actually has a mutation that has changed it to GUG, which codes for valine. And this, of course, single mutation, single nucleotide substitution, results in a change in an individual amino acid, which results in a change in the structure of beta hemoglobin, preventing it from forming that tetrameric oxygen carrying capacity molecule, and results in a missense mutation that causes the disease. And there are a variety of these up here as well. You can kind of poke your way through it and see some of these individual mutants that result in diseases. So these are simple mutations. Now, I should give you a heads up about this, and the key element is really this. If you go to the website Online Mendelian Inheritance in Man, OMIM.org, there is a list of approximately 25,000 genetic disorders in human beings. 25,000. How many of these known inherited disorders can be identified by simple mutation? And the answer is less than 5,000. So approximately 20% of all known inherited phenotypes, and of course, many of these phenotypes are disease phenotypes, how many can be explained by central dogma? And the answer is a very small number. So there must be more going on with the organization of the genome, with the coding of phenotypes and traits by DNA than can be accounted for by all of the mechanisms we've talked to this point. and we'll talk a little bit more about those next week. You guys take care. Hope you stay safe. See you on the next one.